So it's 10 o'clock, and I would like to welcome you all to this webinar. And we have um, the main topic here is avian coccidiosis, and we will have different aspects of this. We have a very interesting program, and I will start sharing my presentation. But I first would like to introduce Natalia Mayo from Barcelona, IRTA, and she is the person that took the initiative to this webinar, and this is a part of the BroilerNet. So I will first give a brief presentation of the
Good morning, everybody, and thank you for attending the webinar.
So, and please, you can ask your questions as I wrote in the chat. You can ask your questions in the question and answer function at Zoom, and then we will bring the questions to our speaker. So the program of today is that we will close this webinar not later than 12. We have 3 speakers. We have Dr. Damon Blake, that is professor in parasite genetics at Royal Veterinary College, Dr. Tiago Prucha from VetWorks, and then Dr. Silvestre from INRAE in France, and they will give their presentations. I think it's a very nice thing to have a short break in the middle, and I know that it's appreciated. So between the presentation of Tiago and Anne, there will be a 5 or 10 minute, it depends on. So we will have a short break there. And then we will have a general panel discussion, and I'm very happy to moderate it. And as this is part of the BroilerNet project, I would like to give you some information about the BroilerNet project. It's a European network funded by the European Union and the European Commission. And it's a network for exchanging knowledge within broiler production. So what we have been doing since 2022 is to build a network to enhance the resilience and sustainability of the European broiler sector. We have 25 partners in 13 different countries, and in all countries we have one industry partner and one academic partner. So the academic and the industry partners are working along, and in this project the industry partner is also funded by the European Union, which is not the case in some of the EU projects. So what we have been doing is to build national networks in all the countries, and then we have created an international network with the experts. We have been fostering interaction between science and practice. We have asked all the broiler innovation net groups in the different countries, in the 13 countries, what do they think is the sector challenges that they would like to have an answer to? And then we have collected good practices within this network, and that's the good practices. They should be ready to use and they should be innovative. And in order to have a clear focus, this is focus on the broiler farm and the farmer. The hatcheries and the abattoirs is, of course, a very important part of the food chain within broiler production, but this has had the focus on broiler farmers. And we have also, in order to structure this, we have 3 themes. We have the environmental sustainability, we have animal welfare, and we have animal health. And this webinar is focusing within the animal health work package, and Natalia Mayo is the work package leader of that work package and that theme. So if you want to know more about BroilerNet, you can find more information on our website. We are at LinkedIn, and we also have videos on YouTube. And if you scan this QR code, you will get to our website, and then you can find lots of practical information, fact sheets, animations, and you will find videos. And after this, maybe it will take a week or two, but then you will find also the today's webinar as a video. So I will stop sharing, and this was a presentation of the BroilerNet project. And now it's a pleasure for me to introduce Dr. Damer Blake, which is the professor of parasite genetics from Royal Veterinary College, and he has been working with bacteria genetics and also with parasitology genetics. And this research includes population genetic analysis of recognized and new Emeria species, development of novel vaccine delivery strategies, understanding genetic basis of host resistance to coccidiosis, interaction between Emeria and the microbiota in the gut. And he is also an editor since 2017 for the distinguished journal Avian Pathology. So please, Damer, it will be interesting to hear your presentation. You're welcome.
Thank you very much. It's a very kind introduction. You make me sound like I'm very busy.
It looked perfect for me. So I think everybody can see it very well.
Great.
Thank you.
Thank you very much. So I've been invited to talk about basic aspects and recent developments related to poultry coccidiosis. There's not a massive amount in my talk on basic aspects, as I figure a lot of that is very well known, but we have a reasonable introduction to go through. So I was going to start off by highlighting what I think we all know, and that is that chicken production is booming. You can see here, this is a heat map of the density of chickens produced in 1961. And as the colors get brighter or more intense, you see there's more poultry. So the US, China were pretty much leading the way at this point, but a lot of other countries like Africa, maybe Europe, were rather further behind. But as we move through the decades, you can see it starts to get much brighter. There's far more poultry. So there's now a wider distribution of chickens of a greater density than ever before. In fact, FAO stats was just updated for 2024 figures, and I see that more than 78 billion broilers were produced in 2024. So if chicken production is booming, then it means that pathogens such as Emeria are more important than ever before from an economic perspective. Now, we actually re-estimated the cost of coccidiosis recently, and I'll expand on this in a minute. But essentially, we saw that the global cost of coccidiosis exceeds 12 billion euros every year, and it's equivalent to 18 cents per chick placed. 2022 was a slightly unusual year where there was a surge in the cost, and this is partly because of the consequences of COVID and the start of war in Ukraine, as we saw food prices increase and the value of poultry. Here in the UK, about 40% of drugs, so that's therapeutics and prophylactics, that are sold for use in UK agriculture are to control coccidia. Now, that's not all for chickens, that's also for pigs, but the majority will be for chickens. It's worth noting this figure is fairly old now, and actually they don't release these data routinely anymore. And the last point is that coccidiosis is regularly ranked as the #1 risk for broilers and sometimes for layers in the US, and that's in an annual survey from veterinarians who work with broilers. So this tells us that Emeria are not going away, and control remains essential. So what's the problem? Well, as we all know, pretty much every chicken that's produced is likely to be exposed to oocysts like this. So this is the infectious unit of Emeria. And in the absence of control, many birds will become infected. And they may suffer severe disease of the upper, the mid, or the lower intestine, depending on the infecting Emeria species. Now, the good news is there are several controls. So there's panels of drugs available, panels of live vaccines that we can use to control coccidiosis. But none are perfect. So we know drug resistance is common, especially to the chemical anticoccidial drugs. It develops rapidly, it spreads quickly. And there's now increasing public opinion and increasing legislation to call for the reduction of the use of drugs in livestock production. And of course, this will impact on use of anticoccidials. We're lucky to have vaccines to control Eimeria, and these are something of a success story. There's billions of vaccine doses sold every year, and there's increasing uptake, particularly in North America. But there are questions about capacity, so whether we could produce enough vaccines to replace drugs. That's particularly so for attenuated vaccines. Where wild-type vaccines are used, there's a suggestion it might associate with necrotic enteritis. And overall, there's a perception that cost may be limiting. So if we think about cost, it's worth thinking about the overall cost of coccidiosis. Now, we developed or updated a model that was produced by Ray Williams, and this looks at costs of control in both broilers and layers. And so you've got prophylaxis, drugs, vaccination, but also therapy when it goes wrong. But the major costs relate to performance. So we're looking at mortality, particularly mortality in broilers, and morbidity. And this is where the big numbers come in. So reduction in broiler weight, increased food conversion ratio are key. Now, we used data from 2016. This was a paper that was published in 2020, and it showed that the cost of coccidiosis in the UK at that time was about 115 million euros. In Europe, just shy of 1.2 billion, and in the world, about 12 billion euros. Just to repeat that figure, that's about 18 cents per chick placed. Now, if we're thinking about control, it's worth highlighting the importance that anticoccidial prophylaxis, so particularly drugs, have played in the development of poultry production. So we can see in this graph, this is the chicken population per year in billions. And in the 1940s, the figures were fairly low, less than 1 billion chickens. There was steady progress through the 50s, 60s, 70s. They then became more industrialized and took off, so that, as I say, now we're close to 80 billion chickens produced each year. Now, crucially, this was supported by control for coccidiosis. So to begin with, we had the sulfur drugs. This was followed by the introduction of the synthetic anticoccidials, and perhaps most dramatically, the ionophores. And of course, ionophores dominate the drugs used to control Eimeria now. It's worth noting there have been no new drugs released for control of coccidiosis since the early 1990s. And although I know there are several groups working in this area, I don't think there's any coming in the immediate future. Now, in addition to drugs, we've also had vaccines. So live anticoccidial vaccines, the first were introduced in the early 1950s. And of course, the attenuated vaccines that we'd be familiar with in Europe were introduced in the late 1980s and the early 1990s. So just a bit of background on those two types of vaccines. So obviously we have wild type or non-attenuated live vaccines, and this means they're essentially virulent. Now, the first of these vaccines was introduced in 1952, so it's a very early vaccine. It was thanks to work by S. Allen Edgar, among others, and essentially it uses low doses of oocysts that are administered to chickens to give a natural infection. As the parasite cycles, the birds become immune. Now, until recently, this was primarily used for breeder and layer stock, but there's been quite a dramatic expansion of vaccination, particularly in the US. And there's two influences for this. The first is bioshuttle. So that's where birds are vaccinated using a virulent vaccine, and that's followed up with an anticoccidial drug. And the second is a quirk of legislation, in that ionophores are considered to be antibiotics in the US. So if you rear a chicken that's antibiotics free, such as no antibiotics ever, it means you cannot use ionophores. That's not the case in the European Union. The second type of vaccine are attenuated vaccines. So these were selected for precocious development. And this was work from Tom Jeffers originally in the US, but it was picked up by Martin Shirley and others to develop a series of vaccines like paracox, and you'll be aware now of Huvigard, Evelon, Hiprocox, a range of others. And the key is they have lower replication, therefore they're less pathogenic and they're safer. But they also have a comparatively limited production capacity, and that makes them more expensive than the standard vaccines or the wild type vaccines. So we had a fairly brief look at the basic aspects of control for coccidiosis. And the reason I've done that is because if I gave this talk to you 10 years ago or 20 or even 30 years ago, it would have been pretty much the same introduction. Eimeria are widely considered to be one of the ultimate enzootic pathogens. We've known about them for almost a century. We have some understanding of how to control them. We understand their epidemiology, and they persist, and they're not changing that much. But I would say that has changed actually in recent years, where there have been some, as the topic of the talk, some recent developments that are quite different and new for coccidiosis. Now, the first of these, which is going to be most of my talk, is going to be understanding Eimeria species. And the second part will be looking at Eimeria populations within those species to see what actually happens on farms. So if you look at this is a field card from the US from 1969. It's actually something of a work of art if you look at it. It highlights the 7 recognized Eimeria species. So we have Eimeria cervulina, brunetti, maxima, mitis, necatrix, praecox, and tenella. But you'll also see 2 other species named here, Eimeria mivati and Eimeria hagani. Now, we know both of these are not valid Eimeria, but if you're in the US, depending on the vaccine you're using, you might see Eimeria mivati mentioned. Now, this card documents where they should replicate in the gut of the chicken, the type of lesions you'd see, the size and shape of the oocyst, the pre-patent period, and a few other variables about their life cycle. And that's been dogma for decades, but something changed in 2008 with the publication of a paper from Australia. Now, in this paper, they were looking at a coccidiosis outbreak in vaccinated broiler breeders in Australia. And they originally thought it was perhaps a vaccine failure because coccidiosis had occurred. But when they started to sequence, and here they're looking at just one part of the genome of each of these parasites, they found 3 completely new sequence types. And of course, this was not really understood. So were they divergent strains? Were they cryptic species or something else? At that time, they were given the catchy name of operational taxonomic units, or OTUs, and they're called X, Y, and Z. Now, we subsequently undertook a survey, and we found that these parasites are quite common in Africa, in Asia, in Australasia, and South America, but it was not really clear whether they're common in Europe and North America. So this is a map of the occurrence of the different Eimeria species. Everywhere you can see a pie chart is an area where we conducted our molecular survey. And every pie chart that has a red slice had all of the 7 recognized Eimeria species. So you can see every country we sampled had all 7 species. If you have a blue slice, that tells you that the new genotype X, or OTU-X, was present. A yellow slice, OTU-Y, and a grey slice, OTU-Z. Now, they were first described in Australia, and so as you would expect, all of these parasites are here. If we look at sequencing data that's been produced in the US, you can actually find details of all of them as well. If we look across sub-Saharan Africa, we can see that many are present. So in Nigeria, they were all found. In all of the others, OTU-Z was found, in most OTU-Y. OTU-X. And the same is the case in Venezuela. When you move further north, so in North Africa, they weren't found. But if you look more recently in Europe, and here we sampled several European countries, we do have 2 of these new genotypes, Y and Z. So if they're widespread, what are they? Why is anyone interested in this? It's perhaps academically interesting, but is it relevant to a farmer? So as a parasitologist, the first thing we do is we look at the size of the parasites, to see how they compare. Now you can see here a scatter plot, and these are the size of all of the recognized 7 Eimeria species that infect chickens, and the 3 new cryptic species. It's difficult to see what's happening, so we can zoom in. You can see OTU-X is a big parasite. It's the same width as Eimeria maxima on average, but it's a little bit shorter. OTU-Y, that fits within the Eimeria brunetti area. And OTU-Z is a small oocyst which overlaps with Eimeria mitis and Eimeria cervulina. So this is suggestive that there may actually be at least 1 new species, as OTU-X is quite different, but it's not really conclusive at all. So we undertook some biological characterization. For example, OTU-Z, we found that schizonts, that's the asexual phase of replication, can be detected from about 50 hours post-infection. We see later generation schizonts between 96 and 168 hours. The parasites replicate in the villi, not the crypts, which suggests they're not highly pathogenic. And we can see gametes appearing from about 120 hours post-infection. And this correlates with the time when we see the greatest pathology. Now, there's no clear lesions, but what you do have is a profuse mucoid enteritis. So we can pool our knowledge of these new parasites and compare them to the recognized species. So, for example, INRAE cervulina, we know replicates in the upper part of the gut, primarily the duodenum. INRAE brunetti towards the end, INRAE tenella in the ceca. We see that OTUX replicates in the upper part of the gut. Remember, its oocyst looked most like INRAE maxima, and it replicates in a similar area, perhaps just a little higher. OTY, its oocyst looked most like INRAE brunetti, but it replicates in a very different part of the gut. That's much higher up. And OTUZ, which most looked like mitis and a cervulina, replicates in the same area as INRAE cervulina. We can also look at things like pre-patent period, so that's the duration of time between infection and the first oocyst being shed. We see that these cryptic species all have long pre-patent periods. Only INRAE necatrix is consistently longer. And we can look at the type of disease they caused. So brunetti, necatrix, and tenella all cause a hemorrhagic type of coccidiosis. The others are malabsorptive form. And that's the same for these three cryptic species. So, of course, using some modern technologies, we went on to sequence the genomes of all three of these new species. So that's OTUs X, Y, and Z. And we're able to generate more than 10,000 gene models for each. Now, using a lot of the genes that are traditionally used for characterization, so things like actin depolymerization factor, beta tubulin, mitochondrial genomes, we're able to put together a family tree. Now, what you can see at the bottom, this is INRAE falciformis. So this is an INRAE that infects mice. And so that's the outgroup. We see that INRAE necatrix and INRAE tenella are very closely related, but they are distinct species. So the length of this branch gives you an indication how closely related they are. We then look at the first of these new genotypes, OTUX. We see it's closely linked to INRAE maxima, but at longer branches than was present for tenella and necatrix. The same is the case for OTY and INRAE brunetti, with OTUZ being particularly unusual, standing out on its own. So combined, this suggests there are, in fact, three new INRAE species that infect chickens. So to help people understand these, we put together a dichotomous key. So we're saying, well, do they replicate in the duodenum or the jejunum? If the answer is no, you can exclude brunetti, mitis, tenella. Do they have a pre-patent period less than 120 hours? If the answer is yes, you can exclude a cervulina, maximum praecox. Do they cause hemorrhagic disease? If the answer is yes, we can exclude necatrix. And we can then just separate the remaining parasites based on size, with the smallest being OTUZ, the largest being OTUX. Now, at this point, we decided to be brave and to actually start to name these parasites. So the first, we called INRAE lata, lata meaning wide. The second is INRAE nagambi, that's the area in Australia where it was first detected. And the third is INRAE azaria, that's an area in Nigeria where we found an isolate. Now, if you think back to this card, this was from the 1960s, remember? There were 7 recognized species, but they also mentioned 2 others, INRAE mivati and INRAE hagani. And the question is, could we have discovered these? Well, based on this card, we know that's not the case, as they have very different life cycles. So we do have 3 new species. But so what, I hear you say, is this really of any relevance? Well, the first thing we wanted to see is, are these new genotypes pathogenic? So taking INRAE azaria, this is an isolate from Australia. We infected Ross 308 chickens, 21 days of age, and we monitored body weight gain. It's fair to say the body weight gain is perhaps not as good as might be expected in practice. We are better at it now. But the key message is that in the presence of infection from 5,000 oocysts, there was a significant reduction in body weight gain. And if you give 100,000 oocysts, there's a further reduction. So yes, these parasites are pathogenic, but there's no mortality, even at this high level of dose. Instead, we just saw a watery mucus from days 5 to 7 and no discrete lesions. So this suggests that there's an economic relevance to this parasite, but it's probably not highly pathogenic. So does it matter? Well, drugs are likely to be equally effective against these parasites. We know that live anticoccidial vaccines are effective against the recognized species, but the question is, are they effective against the new species? Because with drugs, whilst they're effective, they're equally likely to be at risk of developing resistance or problems like that. So to test vaccines, we took two different vaccines, one an attenuated European vaccine, one an attenuated Australian vaccine. We vaccinated groups of birds, vaccinated them 3 times to make the vaccination as strong as possible, and then we then challenged them. So we can see here first, this is vaccination using a European vaccine. We've got birds that were unvaccinated or vaccinated 3 times. And this control group received the vaccine as a challenge as well. You can see the vaccinated birds produced essentially no oocysts. The vaccine worked, but there were a large number of oocysts produced in the unvaccinated group. But if we look at these 2 new species, so Aimyra lata, here it replicates very well in the presence of the vaccine, pretty much exactly the same as in unvaccinated birds. So there's no seemingly protection here. Although if you vaccinate with self, it would protect against self. And the same is the case for Aimyra azaria. Now, when we tried to publish this first, a reviewer commented that we'd used a European vaccine with an Australian strain. So we repeated some of this trial, and this time using an Australian vaccine. And you can see exactly the same results. Again, the vaccinated birds, Aimyra azaria, replicate very well, pretty much the same as the unvaccinated birds. And of course, remember, this parasite was first isolated in a group of broiler breeders in Australia that had been vaccinated. So if we ask, are live anticoccidial vaccines effective against the new species? The answer is no, which suggests there could be a return to reliance on drugs. Now, if we remember this survey, we actually look in a little more detail now. So within Europe, we found Aimyra negambi. Remember, that was the OTUY parasite. In Italy, Romania, and the UK. We found Aimyra azaria. That was the OTUZ parasite in France, Greece, Italy, Poland, Romania, Spain, and the UK. And these countries highlighted in blue are part of a slightly larger surveillance study. We detected this parasite on about 10% of farms. So it is present in Europe, and whilst it's probably not highly pathogenic, it is increasingly common. So we've spoken about understanding Aimyra species, and that helps us to look at populations and species occurrence. But what about subspecies occurrence? What happens within these species on farms? Well, to assess this, we're able to use some new tools. So we've used 18S deep amplicon sequencing. Now, this is very similar to microbiome sequencing, which I imagine most of you are familiar with. So if you think about microbiome sequencing, it's where total DNA is extracted from a bacterial population. So it could be a fecal population, a litter population, it could be water or anything else. You have total DNA. You then use a PCR that targets a gene that's present in virtually all bacteria. You then sequence it tens of thousands of times. And when you annotate each of those sequences, you can look at the presence of different bacteria. So whether it's a different phylum, class, order, genus, species, or even subspecies. And if we talk about a subspecies, we're talking about a particular sequence type or an amplified sequence variant. Now, we've taken this approach and we've generated different primers that target not the bacterial 18S ribosomal DNA, but the parasitic 18S. So we're now able to take a coccidia approach to microbiome sequencing. So what we've done is to apply this in different flocks during rearing to see what happens in the different parasite species over time. So this is an example of parasite counts from a flock. It was a traditional broiler a broiler flock. They were given a normal chemoprophylaxis. There was no signs of disease. They were essentially a healthy flock. We didn't detect any oocysts at our first sample point. Very small numbers, 14 days of age. There was something of a surge, although not to massively high numbers. By day 21, this came down as the birds became immune. And again, the flock performed relatively well. Now, when we apply the 18S tool to this, we could see that at day 14, there were very few oocysts, as I mentioned, but mostly they were Aimyra cervulina. If we look at day 21, again, mostly Aimyra cervulina, but you can see there's maxima present, there's mitis, there's praecox, and tenella. Now, there were differences in the proportions between 2 different sheds, but essentially they were the same parasites. And you see as these numbers go down, you see an increasing importance of Aimyra tenella in the later stages. Of course, we know this parasite has a slightly longer life cycle, is less immunogenic. It takes longer for birds to become immune. So this is pretty much a standard profile. Now, what was interesting is what's happening within these species groups. So if you look at this top one, here we're looking at individual sequence types of INRAE cervulina. You can see that one sequence type was dominant. They all start off at a low level. This one surged through to become overwhelmingly the most common INRAE cervulina, then reduced as the birds became immune. There were a few other genotypes present, but they were all much lower. If you look at INRAE tenella, however, a very different picture. You have one genotype that was dominant, but there's actually, 1, 2, 3, 4, maybe even 5 other genotypes that were also common. So there's quite a complex population here for INRAE tenella, whereas much simpler for INRAE cervulina. Of course, this might have an effect on control if, for example, some of these are associated with resistance to drugs. And we've then gone on to compare these between production systems. So this is the same graph where we looked at INRAE tenella, but now we have samples from a broiler breeder unit. These were vaccinated. Now, the first difference you can see is there's relatively high numbers of oocysts at day 8, where we didn't find anything in the drug-treated broiler unit. That's to be expected. They've received a live vaccine. We then saw that oocyst numbers came down quite dramatically over time. As you would expect, the birds would become immune. And in shed B, they stayed low throughout. In shed A, there was a slight resurgence by INRAE tenella around day 64. But if you look at the actual OPG figures, these were all fairly low. So you would say the vaccine has worked, the birds were healthy, and there wasn't a problem. But it is interesting that the most common genotype from the vaccine was present still at day 64, but the other 2 genotypes below were now different. So this indicates that a field strain has actually got in and has managed to replicate in the face of that vaccine immunity. So to summarize, we can say there's little change in biological understanding of coccidiosis over many decades. The drugs we use essentially be very similar. Perhaps the way in which we deploy vaccines is changing a little. But we now know there are some changes. There may be 10 INRAE species circulating in chickens, or maybe even more. These may pose an unrecognized threat to Europe and North America. We've seen that these parasites are present and relatively widespread. The new species could be detrimental to poultry health, productivity, and welfare. And we've seen that current vaccines are not effective against these species. But it's worth noting the risk or the pathogenicity of these parasites seems to be low. They're slow growing, so they're really more of a problem for breeding stock. Don't seem to be a big problem for broilers. We've also seen that INRAE populations and their genomes are probably much more diverse and dynamic than we'd realized. So there could be multiple different genotypes on a farm at one time. And what we don't know is how common is cross-fertilization between these genotypes, because this is one of the ways in which drug resistance will be disseminated very quickly. And of course, understanding these population genetics will be relevant to the control and the treatment of coccidiosis in the future, which you're going to hear more about in the following presentations. So let's finish up by highlighting there's quite a large team of people who are working at RVC, and they've been behind a lot of the characterization and the surveillance for these parasites. Thank our partners in Australia at Bioproperties who've helped with provision of samples, in Nigeria the same, and the team at the Wellcome Trust Sanger Institute who have been sequencing the parasite genomes for us. So I'll stop sharing there. I think we've got a few minutes for questions if anyone has any now.
Thank you very much, Damer. And please, I can't see that we have any questions. Thank you very much for a very interesting presentation. And I really got an update because my pathology or parasitology knowledge in INRAE is quite old. However, I have a question to open up, and that is that we do you know more about the non-pathogenic INRAEs? I mean, could there be INRAE species that becomes pathogenic and that we do not know about yet?
It's an interesting question. So if you look to the literature, I think INRAE praecox was long thought to be non-pathogenic. But actually, that's not the case. There was a study published from a strain in Wales that found it had a similar pathogenicity to INRAE cervulina. And of course, it's deployed in several vaccines. So It does vary. I think there's a lot of Eimeria that are probably not highly pathogenic, and that's probably why we've not detected them. Because if you've got a pathogen like Eimeria necatrix that's causing bloody diarrhea, then it'll be obvious. But if you've got a parasite that's not got a high level of pathogenicity, doesn't cause clear lesions, and only impacts on weight gain, food conversion ratio, it's very easy to go under the radar. I mean, it's interesting to think where these parasites came from. Have they emerged from some other population, or is it actually they were always there and we just didn't recognize them? And I'm inclined to think they were probably always there. They've been probably controlled by drugs to the same level as the other species. And it's only when you start increasing vaccination, and particularly if you're looking at longer lived birds, they start to become a problem.
Thank you. Anne has a question.
Yeah, thank
Please.
you, Demar, for your presentation. It's always a great pleasure to to assist to your presentation. I have a question about those new species. Did you study a little how they may become perhaps more pathogenic in the context of climate change and increase of temperature? Do you have any idea or signal that they may evolve or adapt differentially than our already known species?
I don't think they'll be enormously different, but we do have some studies in Africa. And actually, I took those slides out because I feared my talk would be too long. And we were looking at in Ghana, in Tanzania, in Zambia. And in those cases, we found that farms that made a positive production, so they made a profit, tended not to have Eimeria zaria present. Whereas parasites that had a negative margin often did have Eimeria zaria present. Now, I'm not suggesting for a second that that's entirely related to that one parasite, but it's certainly indicative of the production system or something of the presence of them. So I think when it's more humid and warmer, all of these parasites we know sporulate better, they survive better. You see in tropics and in rainy seasons. So it could be that whilst it's not different to the other species, something that's not highly pathogenic, if it has a chance to sporulate better, it'll be present at a higher level and it then becomes more pathogenic.
Thank you.
That's a good question.
Thank you. Natalia has a question, please.
Yeah, thank you, Stefan, and thank you, Demar, for this very interesting talk. For your experience, which is the best approach to diagnose this new species of Eimeria? I mean, you mentioned that the lesions are non-specific. So is the molecular tool PCR or something that is specific for detecting these new species?
There are. So what we've been using essentially is PCR assays. And the data I showed obviously is using next generation sequencing, which is more expensive, harder to use.
Yeah.
But there are now panels of species specific standard and quantitative PCR assays available for all three.
Oh, okay. Thank you very much.
Thank you very much, Damer. And for the participants in this webinar, please ask questions and we will have answers. And we will move on, but there will be opportunities for Damer to answer more questions later on in the panel discussion.
And I should say, sorry,
please.
if anyone does have any questions after the event, I've got a fairly unusual name. If you Google me, you should find me quite easily, and I'm happy to answer questions afterwards. Thank you very much.
Yeah, thank you. Thank you. And then we move on to Dr. Tiago Prucha, and he has a master in veterinary medicine from the University of Porto, and he is a poultry veterinarian, and he has been a farm manager for a large integrator in Portugal, and he is now poultry consultant for VetWorks, where he has been working for 7 years across more or less all continents except Antarctica, I can see. And you worked with leading companies, and you were also involved in different EU projects like FagoVet and Net Plus Safe. And you are also a lecturer in the postgraduate poultry health science chair at Ghent University in Belgium. So please give your presentation. We're very eager to hear you. Thank you.
Hi everyone. Thank you for that very nice introduction. I have the very difficult task of talking after Professor Demer, which by itself, it's a very big task. It's my pleasure to be here and to explain to you about the more practical look into prevention and control. I don't intend to do a very theoretical presentation, so my goal is really to look at how are the main points and the low-hanging fruits that we see in prevention and control of coccidiosis, and how can we move towards the challenges that we have nowadays, or through the challenges, let's say, and understand how we can control it better. So more than, let's say, a fancy formulation, we should focus first in these very basic things that will have a very big impact. So in my presentation, I will, as said, explain the different tools that we have nowadays and how they work between each other and what are the things that we should look at. I think Professor Reimer already gave a quite good introduction in terms of how is the disease, or what are the disease challenges nowadays. So no need for me to go over that. But we know that coccidiosis is still quite a big challenge. So in this presentation, I will try to go over the different points that make it so. So we know that a very big part of the success that this parasite has in the industry has to do with the way that we produce our chickens. So this parasite evolved from a place where it had very low density of chickens in terms of population to a very efficient way to cultivate itself. Also, the fact that we reuse the same places to rear our chickens make it so that there's a stacking process over the different cycles. In terms of outbreaks that we have nowadays, a point that is important to stress is that when there is an outbreak, is because something went wrong already. So we shouldn't get to that. Therapeutics are parts of the solution, but are not for sure a long-term solution. So they are, let's say, a fire extinguisher, not a prevention plan. So here the focus is really on the monitoring and understanding how the molecules work, what are the different points that you should take into account, and how you should play with them in order to make sure that you control the, let's say, the less good part of the molecules. So the first thing that I think we should understand before going over the different molecules is to understand why we still have coccidiosis outbreaks. Well, there's never one single factor. So high infection pressure is one, obviously, but all the small failures, vaccination, stress, virus, feed intake will have a role. The immune response as well, but is also linked with all of these small factors that I have. And the one that I will focus more maybe during this presentation is really on the drug itself, so resistance, the way that we use it, and also pathogenicity of the field strains that we heard about already, and suboptimal dose levels that could be either because we have bad application or because there were any mixing errors or selective eating while in the house. So those are different, let's say, reasons why we have it. In terms of tools, it's quite easy because we don't have that many. That's also why we have to be careful in the way that we use them. So let's say we have 3 or 4 categories. Therapeutics will not be a long-term solution, so will always be a short-term solution. And then we have the alternative products that are normally not advisable to use by themselves as a standalone. And then the vaccines and the anticoccidials that will be, let's say, or support tools on the long run. Although there are several programs that don't use one or the two, depending on the part of the world that we are talking about. But in a general perspective, let's say, we tend to use vaccines and anticoccidials as let's say, long-term support tool. And then within the anticoccidials, we have these 2 different tools, chemical products and ionophores. Although ionophores are also chemicals, they vary mostly on the mode of action, which I'll touch on briefly after. So in a very quick mode, just going over the different programs that we have, there are obviously huge variations in terms of programs. In a general overview, this is what we will have. So a dual or shuttle program in which we will have 2 different molecules in during the cycle. So during the starter and the finisher phase, a straight or full program in which we use the same product, and then a bio shuttle, which is like a compromise between both, sometimes with the organic or let's say antibiotic reduction or antibiotic free stamp, but is not in fact antibiotic free magic. So let's say, so there is a compromise here that and it needs to be taken into account. So very quickly over the different tools which Professor Reimer had explained already, but going over them. So the vaccine, it's a live vaccine. So the different vaccines of coccidiosis, they work by replacing the resistant field strain. So basically what we want to do is to potentialize the cultivation of these sensitive vaccine strains in order to increase the efficiency of the programs that we are using. That's why vaccine improves the future performance of the anticoccidials as well, also allows us to play a bit with the type and cost of the products that we use after vaccination. So it's not always a black and white picture. They are not cheap. They are becoming more affordable, but they are still not cheap. So different uses depending on the bird value that we are considering. Obviously, if we talk about long-living birds, it makes it a lot more affordable to pay for these kind of solutions. If we're talking about broilers, it's still not as common as we see in long-living birds. There are different things to take into account when we talk about vaccination. They are usually administered between day 1 and day 7. Different ways of application. They can be in spray, on feed. They can be in the hatchery. They can be in the farm, in ovo as well. Having in account that they are a live vaccine and they have different levels of attenuation, you also have non-attenuated and attenuated vaccines. You will have the potential of having direct problems with bacterial enteritis and or necrotic enteritis, depending on the part of the world where you are and the outside factors as well. Because they will have to reproduce themselves, so there will be a damage to the enterocytes. And so here it really becomes important that we give the support to the gut when we give this vaccination. Very important to understand when when we reestablish, let's say, the accessibility to the products that we are doing, how do we do that? Several ways to assess that, but the most common way is by having a regular monitoring in which we look into the OPGs and we see where it spikes. When this spike, let's say, goes to a younger age, it means that what we are seeing is the vaccine strain and not the field strain. But there is, of course, other things that we have to take into account. For instance, escape of immunity and other problems linked to the fact that it is a live vaccine. So here I have just a study that shows the impact of Cox vaccination. The biggest benefit is not obviously FCR improvement, and most of the studies that you will see will have actually a worse FCR, for instance, when during vaccination, but after is where we see this stability and predictability in terms of molecules that we can use. And here in this case, we looked not only into the FCR, but also at antibiotic cost, in which we saw a huge decrease in them, and it's quite a big sample in which we looked at, but that will be the main thing. So an example of what could happen in practice in terms of return on investment and how most of our customers look at vaccination nowadays. Very obviously, very simple calculation, but just to show the importance and the efficiency that vaccine might have. So here in this case, we have two different scenarios. One very standard program in which we have narasin, nicarbazine from day 0 to 25, and then narasin from day 25 until withdrawal, and another case in which we have vaccine. So in one case, we have, obviously you have variations in prices and so on, but this is just to prove a point. In one case, we have, let's say, 30% higher costs with the vaccine, and with the standard program, a lower cost. So if we look at the reductions, let's say that we have with the vaccine, we will see that from the bat, we will have €6 per 1,000 birds less antibiotics, so that we don't use. And then the net cost during vaccination itself will be €4. So that's let's say the difference between the two of them. So at the end, what we have is €6 less of use of antibiotics. We have a 3.5 point gain in FCR, so that's about €25 per 1,000 birds. Again, this will vary, of course, depending on the market that we are. And then we can use, because we increase sensibility of the strains that we have in the house, with the vaccine strains of about, we can reduce for a cheaper program as well. So let's say that we put everything together. So we are talking about €51 versus these €4 that we have in terms of cost, which is the difference between one program and the other. So it's about 12.75 return on investment if we do the math. So this just to show the return on investment. So in terms of coccidiosis vaccination in ovo, it's really about looking at timing here and the correct timing to ensure that the oocyst really reaches the gut via the amniotic fluid. The advantages over spray are, you will make sure that you'll have an uniform dose and obviously an early immune priming as well. There's also some risks, obviously, using this, but it's more and more a solution that is becoming more reasonable, especially in cases, for instance, in where we have on-farm hatching, in which this becomes a lot more relevant than in other cases in which we can administer in the hatchery. So very briefly here in the vaccine. So why the vaccine does not work? Well, there are several cases. Again, here the same. There's not one reason why. The shelf life is one, obviously. The high infection pressure that we might have in the house, poor cross protection to field strains as well. We heard from Professor Damer these new INRAE species that we have that obviously play a role in terms of vaccine efficiency, and very often quite far genetically to what we have in the vaccines. And then obviously inability of the bird to develop immunity by stress, mycotoxins, virus, different things that might play a role here and that can cause an impairment of immune development. So how many cycles before restoration? There's not one number. Here it really depends on the field strain, the challenges that we have, the infection pressure as well, and when we have how many cycles we need to restore this sensibility, but hard numbers between 2 to 5. The easiest way to do it is really looking at the oocysts per gram count and to assess it in a regular way in order to create a link between what we see and what we do. So between the different solutions that we have, I'll touch then on the anticoccidials and chemical products. Ionophores are in some places considered as antibiotics, although they are not antibiotics. They do have an antibiotic effect, which is not a bad thing at all, because it also has the ability to give us some base in terms of bacterial enteritis control, for instance. There are several countries in which we cannot use ionophores. In Europe, we are still safe. Let's see for how long, but we are still safe to use them. But here, the important point to mention is really that they are under pressure, and the efficient has to be safeguarded. Also, because in the last few decades, we haven't seen many other products being developed. So it's really important that we have that in account. So these anticoccidials, they are used for different animal species. Vaccines, they are only used for certain species. They work against all immediate species. Vaccine, not really. The ionophore, important to mention that we do have this favorable side, so we can play with it as well. The important thing here is to avoid reduction in sensitivity and resistance. We haven't had, as I said, no new active developments or let's say relevant ones that can really play a role here. But their strengths are really the broad coverage, the predictable effect, and the fact that we know them quite well. Here, rotation ends up being the main survival strategy for these products, because it will be the only way that will give us this the way to maintain it for a longer term. So you have, let's say, 3 different categories, although it's 2 main ones: ionophores and chemical products. As I said, they are both chemicals. Important to say is that ionophores, they will have 3 different classes, in which between these different classes, the mode of action is very similar. So the risk here is really with cross resistance. So the fact that between these groups, we will have a big risk of cross resistance. On the chemical products, it's not the same. Also, the way that they work in terms of sensibility is also different, but most of these products will have a different mode of action, which means that, okay, we don't have to worry that much with the rotation between them. We do have to rotate between the same, between different products, just not to use the same product. And then we have a combination in which we have ionophore and chemical in the same product. So what are the issues with anticoccidials? On one end, we look at sensitivity resistance, on the other cross resistance, which is more linked to ionophores, and the reduced sensitivity resistance is then the fact that we use them for a long period and we increase the risk of having this increased reduced sensitivity. So what happens is that after a while, we tend to select, let's say, for higher resistance strains, and which will then lead to these cases of resistance over time. There are also other things that we need to look at. I'm sure that most of you have seen this picture in which we have two different scenarios, one where we use a chemical and the other one where we use a ionophore. In one case, we have less efficiency, let's say, in which we still have some coccidial leakage, which is the case with the ionophore. And in the other, we have a chemical in which we don't have any coccidial leakage. It's easy to say that obviously the scenario that we want will be the chemical, but in fact what happens is that the ability to select for higher resistance strains is much higher on the chemical than on ionophore. So although let's say ionophores allow some coccidial leakage, they also decrease the resistance over time. So when we are looking at resistance or efficacy, let's say, we see something like this. So we start from a higher efficacy, although over time, the resistance of efficacy is much higher on the ionophore than what we have on the chemicals. So the key is rotate between the different classes that we have, not only the ionophores, between ionophores and chemicals, but also between the different classes of ionophores, and then using the vaccine as key to, let's say, reestablish sensitivity. And then a bit on cross-resistance, which is really a problem more linked with ionophores, in which we have these different or these similar modes of action. So it's really the point is really to look at the way that we use these different classes of ionophores. So we have these three different classes: monovalent ionophores, divalent ionophores, and monovalent glycoside ionophores. Now, obviously, it seems very complex because I'm explaining it in a very quick way, but it's easy to understand, in which you just have to rotate between them and make sure that we don't leave it a longer period. So something to point out as well is the importance of cleanup. Because chemicals have this higher efficiency, they will allow us to reestablish, let's say, or to clean up the field strains that we have circulating. So before vaccination, and to reduce the challenge that we have for these vaccine strains to reproduce themselves, is important that we understand the value of using a chemical before vaccines. So at the end, what we will see in a normal cycle will be something like this. So this is the oocyst count over time. We have here a threshold in which we start to have subclinical coccidiosis, and here, let's say, is the clinical signs which we don't want to reach. So what we want to do is to allow slowly for the oocyst counts to build up, and then to use the chemicals as the tool to reduce coccidiosis pressure, and then use the vaccine as let's say, the reestablishment of the sensibility that we want to be allowed to use a cheaper program, a less efficient product, whatever it is that allows us over time to work with a long-term solution. So in conclusion, all products have advantages and disadvantages. It's really important that we understand how to play with them, and using this rotation, disadvantages are really minimized and advantages are maximized. This is the way that we want to play with. So this is the overview over the different tools. I'm sorry for this last part, but I was looking at the time and I was seeing that I was short already, and I didn't want to overspend my time. So I think we have now some time for questions as well. Still, I'm happy to answer them.
Thank you, Tiago. Natalia, do we have any questions in the Q&A?
Yeah, there is one question that actually are three questions related. So, are vaccines used alone, or does most breeders use them in combination with phytogenics?
Yeah, as I showed in the presentation, so vaccines will be, coccidiosis vaccines are a live vaccine, meaning that they have to reproduce themselves. So it's a live parasite that is there. You have different levels of attenuation, and even between the attenuated vaccines, let's say you have some vaccines that are hotter, let's put it like that, than others, meaning that they will have more potential to lead the bird into a bacterial enteritis problem or necrotic enteritis problem. So phytogenics are really key to use when we talk about coccidiosis vaccine, because they will give the support to the bird to be able to reproduce these immediate strains without having a lot of gut health problems.
Okay.
Thank you.
And another question that is related to this one is, when used with phytogenics, are the phytogenics applied for all feeding phases?
Sorry, can you repeat?
When you use phytogenics, are these applied for all feeding phases?
Depends very much. When we say phytogenics, it's a very broad range, so it's a bit unfair for me to give that answer as a yes or no. In general, I think that more and more we see the value of using these tools throughout the whole cycle, not only for the coccidiosis, but for multiple reasons. They have immune boosting abilities. They have support or ability to support the bird in other ways and to reduce the use of antibiotics. So in general, I would say yes, but depends very much on the product, the ability, and the money that we have to spend as well.
Yeah, and then another question, the last one on phytogenics, is from a cost perspective, does it make sense to use vaccines with phytogenics?
Again, very unfair to answer with a yes or no. In general, vaccination is really a long-term investment, so it can never be something that we assess as a short-term, meaning that it will never be a cheaper solution on the short term. So if you want to do it well, and if you want to spend some money to make sure that you spend less eventually, or if you have better results eventually in the future, I would prefer to create the conditions for my birds to have as less challenge as possible. So absolutely, yes.
There is a final question, and I think then we will have a break, and you will get the opportunity later. Is there any vaccines available or being developed for in ovo use in broiler breeders?
Yeah, absolutely, yes.
Yep. There are.
Yeah, Heather, I think in my slide, which I will share with you, I have a couple of them as well. And maybe I went through them quite fast, but they are
Yeah,
there.
I think you answered it. I decide that we have 5 minutes break now, and then Anne will give her presentation. So please be back at 11:15, because I promised you a short break. And fill in with some more questions, and we will try to answer them in the panel discussion at the end. But we will not be closing this webinar later than 12 o'clock. Thank you. So be back at 15:15. So welcome back all participants and listeners and to this webinar by BroilerNet on coccidiosis and Eimeria and related topics. So it's a pleasure for me to introduce Dr. Anne Silvestre from France. She's from the INRAE, which is the National Institute for Agriculture, Food and Environment, and her focus is on the host pathogen interaction in context of intestinal health of poultry. And she has specifically been interested in the virulence factors produced by Eimeria and signaling pathways, and also objectifying the anticoccidial properties of natural extracts like plants, seaweed, insects, and possibility to identify active compounds that can be used here. So please, Anne, I'm curious.
Thank you.
Yeah, thank you.
So let's share the screen. This one, I guess. This one. Is it the good one? Stefan, can you see the...
It's in your presentation mode, so we see the first and the second
no,
picture.
sorry. I did not choose the good one. It was this. We made the test just minutes ago.
So,
and is it the good presentation now?
I think you can change it into just that you just present one at a time.
Okay, it's not the good one, so I've got to put this one. I think it's okay now.
Yeah.
And
perfect.
That's okay.
Yep.
So, okay, thank you. So it's a pleasure to be invited to this webinar. So thank you for the invitation and the kind introduction. So it's not easy to have an optimistic message with the control of coccidiosis, but let's try to do this today. I will put my timer on. Okay, that's it. I wanted to take the opportunity to present you the team Be Happy, in which I work. It's headed by Sonia Lacroix-Lamandé. And we study two pathology, the cryptosporidiosis and the coccidiosis, that are disease of the youngs. And we focus on both the mucosal immune response and the host pathogen interaction, altogether to improve the stimulation of innate immunity for the control of the disease and possibly identify new druggable targets. So as it was presented earlier by Damer, there is an increasing problem in the coccidiosis that was previously controlled or that is controlled either in the laying hens by vaccines, and it's my idea was it was quite efficient, but Tiago said that it's not so efficient. But it was efficient to control the coccidiosis in the laying eggs hens, but it's very expensive. And as presented, Damer, the development, the identification of new species may also be a dangerous point that may reduce the effectiveness of those vaccines. For the broiler, most of the control is relied on the use of coccidiostats, but as it was already presented, it was threatened by the development of resistance. So it was just a short illustration of the day, the year at which the molecule appeared at the market and the first record of resistance in the field. And as you can see, sometimes it was as quick as 1 year of usage of the anticoccidia. So in this context, it's more than urgent to find some alternatives. But I think that the term is quite strong And we have to keep in mind that it's very difficult to develop as good as control method than vaccines and anticoccidial compounds that are available today, with the diversity of the approach we are developing in the labs. So I will rather speak about the complementary compounds than the real alternatives to the existing control. And another part is to identify also new therapeutic targets that may be very efficient to control the disease. So the 4 parts of my talk today are divided here in the first, the diversity of the alternatives, and I will then focus on what we do in our lab and how the use of a new platform for the screening may be efficient to study new repurposing drug or a new drug target from the research. So I prepared my talk, and just yesterday I had a literature alert in my email that presented this publication by Flores and collaborators, and it's a very exhaustive review of the subject on the alternatives to the control of coccidiosis, and it starts with the phytochemicals and the botanical extract, and it moves until the very engineered vaccines and methodology to optimize the vaccination of the birds, moving from some nutrition, complementation, and microbial and gut modulators. So I invite you to have a look to this very nice publication, so in animals. And I'm not a co-author, so it's not a self-promotion of the production. And I will just focus on this first part of my talk to several examples that were listed in this publication, and to be short and to keep with my time for the talk. So the botanical extracts are a very nice source of diversity of compounds, and it has been used in a lot of disease control and also in coccidiosis. Most of the active compounds identified are in the family of the saponins, the tannins, the polyphenolic compounds and the flavonoids, and also the polysaccharides. And the action of those compounds may be as efficient for the performance of the animals, and as efficient for the more direct antimicrobial activity. The problem on this kind of research is that you've got to compare some very complex extract full of molecules and more or less active. And it's very difficult to make a correlation between what you can observe in a screening in a lab conditions, well controlled, and what you may obtain in the field situation. Another kind of molecule that may be used are the nutrients and the trace minerals. In fact, the coccidiosis will alter the capacity of the birds to have a good digestion and to profit from the feeding. So it's a way also to support the chicken resilience and to help in recovering from the coccidiosis. Another point very popular also is the fact that the INRAE infection is going to disturb the microbiota composition. And in this context, we know that some Salmonella and Clostridium may increase because of the INRAE presence in the gut. And some study with the probiotics have been intended to maintain a healthy intestinal tract So several compounds have been enriched and have been tested, such as the yeast extract and also the live bacteria. And now we are also moving to a new level with the symbiotics, where several compounds efficient are mixed together. And also another approach is to use the metabolite itself to favorize the increase of several bacteria phylum to compensate the dysbiosis caused by the coccidiosis. Another strategy is also to host target the strategy of the treatment to activate the specific immune responses in the birds. So one strategy is to use the host defense peptides that are also called antimicrobial peptides that are effectors of the innate immunity. In the chicken, we've got several categories like the cathelicidins and the beta-defensins, the ovodefensins. To be honest, I worked with some beta-defensins. So in my hands, as you can see in the publication here, the impact of those peptides against Emeria was quite moderate. When we go to the in vivo data, we can see that during the infection, those kind of peptides are not so modified by the infection. So we may think that they haven't got a real anti-Emeria impact, but more an immunomodulation effect. Another strategy also is the production of recombinant vaccines. So it may profit of the stimulation by cytokine. So you introduce in your chicken some recombinant cytokine, and you potentiate the effect with some host defense peptide to stimulate more efficiently the immunity of the chicken. The recent genome editing production of Emeria tenella strains is also a way to optimize the delivery of the antigen, and you may make a recombinant Emeria tenella strain that is going to express some antigen from other species. In the cases, it was Emeria maxima. And you may produce a super vaccinal strain that would have a protective effect against several species. Perhaps this kind of strategy may be relevant also in the context of the new species that were identified and illustrated by Demer, that if we present a new Emeria species that has a cargo of several antigens, we may have something that would stimulate immunity efficiently. So to move to the other part of my talk, I would like to focus on, if we want to develop new approach, what are the stages that are tractable for the experimentation? And when we want to make a screen of some approaches, what can we do in the lab? So on this picture, I summarized in blue all the stages that are tractable to in vitro experimentation in the lab. So it deals with the first schizogony, and you've got the sporozoite, the schizont in nature and nature, and also the production of the first generation merozoite in the supernatant of the cells. But if you want to access to later stage during the infection, so the second schizogony and also the reproduction, the sexual reproduction of the parasite, you still depends on in vivo experimentation, and we need the chicken to produce the second generation merozoite, the gametes, and the oocytes that we can purify non-sporulated and make the sporulation in in control condition in the lab. So with this kind of stage that are accessible, we can test in the laboratory some compound that will have an impact on the sporulation. We can test the capacity to inhibit the sporulation. We can also work with live sporozoite and determine the effect of parasiticide compound. And with a switch of the color, we can make the staining of the dead parasites. And in a system where you really have in culture some avian cells, you can make the infection, and so you can study the invasion. So you can test for a compound that will inhibit the invasion process of the parasite. And if you make the first schizogony, you can also quantify the impact on the development of the parasite. So taking advantage of the genome editing of Emeria, we made a recombinant strain, two recombinant strains that express the nanoluciferase reporter gene under different promoters, either a promoter very precocious that is easy to quantify the inhibition of the invasion of the cell by the sporozoite, or a later expression promoter that increase in the schizont, and we make the quantification of the schizont in vitro in presence or not of the compounds. All the workflow, in first intention, we work with the global workflow because we can have the impact, cumulative impact of the compounds on the parasite. Then we put the parasite and the compound on the cells, and we make the 3 days development to access to the whole inhibition of invasion and development. And using this kind of platform, we worked with several private companies that were interested in testing for their compounds. Here it's a project Alga Health we had with the Olmix company, which they take from the sea the algae, from the green algae, red algae, and brown algae. And in their project, we made the whole evaluation of the compound on the viability of the sporozoite, the invasion, and also the development. It was the cumulative effect. We had some quite nice results in vitro. So we reached the 50% inhibition, which is, I think, the minimum you have to reach in vitro to think about anything afterwards. And from those results, we think that the future of the analysis will be to make some subfractionation of the extract to decipher if we've got some compound that act more on the parasite, more on the invasion stage, or on the development. In this project, we were asked by the company Phytosynthese to test for several kinds of botanical extract. There were some water extract of the plants and also some essential oil. And it was, the question was different. They wanted to make the characterization of their extract against 3 different steps in the cycle of Emeria. So we quantified the impact of those extracts on the sporulation, on the viability of the sporozoite, or on the invasion. And the project was very useful because for the company, they were able to make optimized mixed of compounds of extract that would act a little on the sporulation, a little on the viability of the sporozoite and in the invasion. And we may have a synergy of those extracts in the intention to develop some feed additive for the chicken. In this project, we were asked to test for the insect and the interest of black soldier larvae in the nutrition of the chicken. So the whole project was very complete with the nutrition and also the health aspect of those extracts. And we compared two types of extract from the larvae on the lipid composition or on the protein enrichment of the extract. All the procedure was the cumulative impact of the sporozoite, the invasion, and the development. It was the whole workflow we evaluated. In the first batch of the extract, we had quite moderate inhibition of the development of the parasite, and most of the results were obtained with the lipid fraction of both extracts. And in a second assay, we made a sterilization of the extract that were filtered. And in this case, the impact of the phenotype was quite low. So it's already an information to think that in a whole likelihood, very small compounds were present in the non-sterilized extract and were efficient in reducing the development of Flameria. So to make a short take-home message on this part of the talk, all our experience support a very large variability of the extract in their composition and a very large variability in their impact and their effect when we test it in the lab. And this is really a real problem in each time we have to work with a very complex extract. They are composed of hundreds of compounds, and it's also a choice to have some quite raw extract that will have a moderate activity, or try to enrich in fractionation of the extract, have a greater impact, but more expensive production of the extract. Another point also is the difficulty to confirm the observed effect from in vitro to in vivo translation. And it's also because you cannot extrapolate from one plate to a chicken. And another point also is a very important point to keep in mind, that the active compounds, the tannins, the polyphenol, they may be anti-nutritional compounds. So it's always a balanced effort to have an impact on the health, but still not reduce the performance of the chicken. So I will finish with a very 2 or 3 slides about the drug repurposing. In the antifungal compounds, itraconazole is a molecule that targets sterol biosynthesis pathways that is quite well conserved between fungus and some parasites. And it was here on the left diagram. We've got a very nice impact of the itraconazole to inhibit the development of Emeria, and making some a collection of derivatives compounds from itraconazole it was possible to reduce the cytotoxicity of the compound and to increase the solubility and possibly to increase the effect against Emeria development. So this kind of strategy may be quite interesting for the private companies because it's more safe on the financial aspect. The market is also more easily to be reached, and return on investment, etc. It's more easy to start with already available drugs. Just to share this information, the itraconazole was target a protein, the CYP51, in other fungus and parasite, and this target is absent from Emeria. So even if we've got a conserved efficacy, the mechanism is different, and it may be very interesting to study this point. Another point to repropose the drug is to take advantage of the discovery of the mode of action of already available anticoccidial compounds. This is the case for allofuginone that was recently published. And knowing the target, you may produce some derivatives and to optimize the production of new compounds. And this is the first step to perhaps develop new compounds to bypass the resistance against allofuginone in the field. The last point is the study of the host pathogen interaction. That is the very conventional way to identify some virulence factors and some druggable target. The ideal candidates has to be essential, druggable, and if they are unique, not present in the host, it's a very good point also. And I would add the early expression of those proteins, because if you try to stop the gametogenesis, for instance, all the pathology already occurred in the chicken, so it may be too late. Candidates that fulfill those characteristics are the calcium-dependent protein kinase and also the ROP kinase that are specific to the apicomplex parasites. Some compounds have already been developed, but it's still difficult to move from the publication and the research in the lab and to find something efficient in the field. And for the ROP kinase, which is really my subject research in the lab, the idea was to develop some peptide that may inhibit the interaction between the ROP kinase and the protein in the chicken to restore the capacity of the host cell to get rid of the parasite. In fact, the infection of the cell by a parasite is going to induce the apoptosis. But Emeria tenella produce a kinase that is going to phosphorylate the p53, which is able to inhibit the apoptosis. So our strategy is to inhibit the interaction between the ET-ROP1 of the parasite and the p53 of the chicken to restore the capacity of the cells to enter in apoptosis. And this kind of research will take advantage of our platform to screen the different peptides we may produce from in silico prediction. So just to conclude, yes, the natural extracts and synthetic compounds library may be very, very, very variable sources of anticoccidial compounds. The parasite-specific proteins are also full of opportunity to identify new therapeutic targets, but it's still a very long way to produce efficient commercial products from the result we have in our lab. The gap between the in vitro and the in vivo evaluation, perhaps the organoids will be a first step to try to fill this gap. The gap also between the complex natural extract, quite raw and very pure active compounds. the problem of the scale-up production of the compounds, and last but not least, to stay economically relevant to the chicken industry. So I wanted to thank my colleague, all the supports I have for my research, and Mamadou Diallo, who worked on the Eat Europe 1 project, and Noé Lemairer, who is working on the peptide synthetic approach. And this is finished for me. Thank you.
Thank you very much, Anne. I think we have there are some questions, and I think we take the question that is specific for you, and that is how does the in vitro invasion model works? Are you determining the rate of the sporozoites that are internal inside the gut cells?
Yes. The point is to wash very well the well to get rid of the extracellular sporozoite, and also the choice of the promoter was important to have something that is more relevant to intracellular sporozoite, conversely to extracellular sporozoite.
Damer Blake will soon get back to the meeting here or the webinar, and we had a question about the people leaving, and this webinar will be available as a video at the BroilerNet website. I can't promise a date, but it probably will take a week or two to get it in proper shape and then put it on the website. So it will be available, and then we can start the discussion here. And I know that there are some questions mainly for Tiago. Natalia, could you select some of them?
Yeah. Yeah. There is a question for Tiago, but maybe also Anne can answer, or they can discuss. There is a question that says, I fully agree with the fast development of resistance against chemical coccidiostates due to their simple mode of action and therefore making them not a sustainable strategy. However, with ionophores, this is not the case due to their more complex MOA. For example, I have several clients that use for more than 20 years the same ionophore without rotating, and where the program is still highly efficient. Maybe the lack of new products developed since ionophores are on the market is due to the fact that resistance development isn't really an issue with ionophores. So, I mean, feel free, Tiago and Anne. I think this is a very hot topic, no?
Yeah, indeed. Well, the first point that I think we should think of whenever we are discussing anything in poultry, actually, is that when we are talking about animals or biology in this case, there's never a black and white answer. I would like it very much to be so, but I don't think there is. And to answer your question, I have indeed people that use the same product for many, many years, and they don't have there are two answers for that. One, they really don't have a problem because management is very good, or because challenge is very low, or because humidity is very low. So the conditions outside and challenge are not created, so they are not there. So it's not per se that the product is good, but that the challenge is very low. And the other one is that they might have a problem, but they don't know they have it. Because monitoring is not that good, because they don't follow it as they should. Of course, in my experience, I think it's always good to rotate, and I've seen plenty of situations in which not doing so has very high costs. But your question is very reasonable, or your point is very reasonable, and I've heard it very often before. And I still believe that the risk of using the same product is much higher than the advantage. I don't know
Please,
if I.
Anne. Yep. Thank you. Thank you. Please, Anne.
Sorry, I had a question to Tiago. I haven't got a very clear picture of what is the situation in the field. When we make some recommendation about the rotation between anticoccidial and vaccines, what is the relevance of this kind of recommendation in the field? Who are the producers that are really switching from one strategy to another? Because I had a quite unique solution for the broilers, for the eggs production, and yes, that's it.
So you mean what type of what are the type of clients that?
Yes.
Yeah, it's obviously much better. You have two different ways of producing chicken nowadays. You have fully integrated market, non-integrated market, and some places where you have a middle term. And it's obviously much more easy to use these kind of approaches in an integrated market because the decision relies solely on one, very often one person. So the efficiency of any decision will be obviously much better. And it's not only that, is that when you have one player, it's much easier for them to be completely honest in terms of what's happening, what are the challenges, in which when you have a non-integrated market, honesty is not always the best commercial decision, let's say. Obviously, it should be, but it's not always the case. And what I want to say with this is that the ones that move towards that are very often integrated customers, although you have exceptions, obviously.
Thank you.
Thank you. Damer, is there anything you would like to comment on this, what Anne and Tiago has been talking about?
I think I missed the beginning of it. It was specifically on what topic?
My question?
Yeah.
Yeah, my
Yes.
question was how relevant was the recommendation to make some rotation between anticoccidial and vaccines from scientist part, and what was the relevance in the field? Who was subject to make this kind of rotation? Because my idea was a clear cut. But thank you. Tiago explained this.
It's not an enormous amount to add. I mean, it is clear in the US they're doing a lot more rotation. There's a lot more uptake for vaccines, and of course you have then the completely different setting in Norway where there's very little in the way of drugs going. It's almost entirely vaccination. So I think what will be interesting in the next couple of years is to see how the parasites evolve, whether you start to see lower resistance in areas that have removed the drug pressure, which the expectation is that's what should happen. But whether that's real, I was just looking back through the answered questions and I see there was one comment about ionophores and how long their efficiency lasts for. And of course, the key to that is that they are complex, but also that they're not complete selection for resistance as well. So you can have susceptible parasites that survive at a low level. So I think in that case, it carries on for a long time potentially.
Natalia, would you like to pick another question?
Yes, there is another one here. They say, as I understood, when using ionophore, there is leakage of oocysts. Even though it contaminates the environment, can we say that it may play a double role, decrease the number of oocysts, but also indirectly vaccinate the birds? Couldn't that be a good thing? So, open question.
Yeah, Damer, do you want to?
So I say that there's some leakage when there's ionophores. Was that the question?
So what I mentioned during my presentation is that there's higher potential of leakage with ionophores than with chemicals.
Yeah, so exactly. No, I mean, I think that's part of their success. That's the reason that one reason they're so popular, because it does it slows development of resistance, but it allows the development of immunity to field strains. So absolutely, I think it's a key part of the success of ionophores.
Yeah. From my side, I understand the question, and the revaccination part is obviously why, for instance, we advise so much to use ionophores after vaccination, because we want to potentialize the efficiency of the vaccine as well. But there's obviously a good scenario and a bad one. If you have very pathogenic strains, you obviously don't want this leakage to happen, obviously. Or in a case in which you are using it for a long time and you decrease the efficiency over time, or selected a bit more the strains, you also don't want this good side. But in general, I agree with the
Yes, I see the question now that's come through. So I agree with Francoise. I think that's exactly the point, that it does indirectly vaccinate birds, and that is a large part of the success of ionophores.
Okay, there is another question for Damer. What would be your advice regarding treatment or prevention towards next flock in case you diagnose one of the new Emeria species? So far, you only can focus on good cleaning and disinfection during downtime.
So our expectation is that the new species are just as susceptible to anticoccidial drugs as others. In fact, we've done some studies which we haven't published yet with Eimeria zaria, and we've tested them against a range of ionophores, and that they perform exactly as you would expect. We have actually most of our isolates are susceptible. We've got one isolate that is resistant to Gunnarson, so pretty much treat them as you would expect other Eimeria. It's also worth noting that they're not highly pathogenic. So actually, if you have low levels, it's a bit like the question a minute ago. They'll be vaccinating the birds and it's not really a problem. The challenge is if you've got birds that you're keeping for longer, so whether they're laying birds or breeding birds, and if you have perhaps a late introduction. And in that case, they behave a bit more like Necatrix in that they appear quite late on, and you can start to see little bits of blood in the feces and so on. But in that case, a normal program for anticoccidial drugs should be effective.
Okay, very good. And then the last question is, I think for all the panel, is there a will from governmental agencies to phase out ionophores? What do you think? Can you answer?
Certainly from the UK. I mean, I think there has been a drive in the popular press. So we have a program called Countryfile, for example, which had an expose about the hidden mountain of drugs being used in farming, and they were referring to ionophores. But I think there is a lot of understanding as well about the importance of these drugs. So at the moment, there's no active drive in the UK to actually rule them out. But I think there is pressure, and in the years to come, there may well be increased pressure. But certainly not a direct threat at the moment.
Yeah.
I fully agree with that. There are places in which we are actually seeing the opposite. And I saw a question there regarding the fact that the US are reusing that. And I think that shows that the reassessment of the importance of these molecules is being considered, because we see that they are still very valuable as a solution, and the potential damage or the potential antibiotic resistance, if that's really a risk, that can bring versus the advantage of using it as a control tool is much higher. So I actually think that in many places we are seeing the opposite, and not that pressure from the government.
Thank you very much.
We had no antibiotics ever, and it's moving now to responsible use, I think, is the switch. And yeah, I think that's going to become much more widespread.
That was a good last word, because we're very close to 12 o'clock, and I promised to finish this webinar by then. So I just would like to add things that you can find in a couple of weeks. Or 1 or 2 weeks, you will find this webinar as a video at the BroilerNet website, and it's broilernet.eu. And we have previous videos from avian influenza, for example, that might be interesting for you. And we also would like to say that we have a final conference on the 8th of May. It will be a main conference. We will have a hybrid, and it is possible for anyone to link into this and be present at video. And there you have the QR code for going into the BroilerNet website. And I would like to thank all the speakers. Anne, thank you, Tiago and Damer. And last but not least, I would like to thank Natalia for organizing this and keeping the track of the questions. And it's 12:01, and I thank you very much. And I see also that there have been people that have a problem to log in, didn't find the code, and they will be able to get in touch with the knowledge from our experts and see the video over and over again if they want to. So a final word. I was thinking of that the Chimera and the chickens, it's like the police song. Every step you take, I will be watching you. We will still continue with this. It's they have evolved together. Interesting topic, interesting presentation, interesting questions and answers. Thank you very much. I hereby close the webinar.